Okay, hello, welcome back. So, in our last lecture, we discussed innate defenses, which are the ones that are non-specific, always respond in the same way, regardless of pathogen. Now, what we want to look at is specific defenses, and these are always particular, specific. They're always going to be defending against something very specific. And what do I mean by that is a particular bacteria. I don't just mean bacteria, I mean a particular species, a particular virus. See, part of the reason why these novel viruses, like the novel coronavirus, COVID 19, is such a big deal for a lot of people is we have no natural immunity to these novel, newly formed viruses that have now come from animals. And it's usually where the most nasty bugs have come from is animals, like COVID 19. So there would be no sorts of natural immunities there. So this involves the action of lymphocytes. Your B cells and T cells specifically are going to be the big role players here. Now, T cells, we do cell mediated immunity. They might be going in for a cell infected with a pathogen, so it defends against abnormal cells and pathogens or inside of cells. So let's say you have a virally infected cell like COVID 19, then these cells can destroy that cell. And again, I was just kind of describing this like Kobayashi Maru. For those of you guys that have never seen Star Trek movies, in Star Trek they had the test that could not be passed. It was an impossible to beat situation, and that was the point. Is they had to go through it, but Kirk cheated and reprogrammed the computer and everything, and beat the Kobayashi Maru. But anyway, because he didn't believe in no-win situations. So if you have a cell that is infected, we often will destroy those cells. We'll sacrifice the needs of the many for the needs who outweigh the needs of the few. Now B cells, this is antibody-mediated immunity. We call this humoral immunity because this takes place. The fight occurs in your body fluids like the tears, the saliva, the blood, those kinds of the interstitial fluids. These are the fluids in the body called humor, and there is going to be an antibody, very specific antibody for a very specific antigen on a very specific pathogen. So not only would it be, if that specific pathogen was COVID 19, it's going to be one protein on the surface of COVID 19 that we would have to have produced immunity to. And this is how Jenner created his very first vaccination, was that the proteins found on the surface of cowpox. There was a protein that was also found, identical to the one found in smallpox. And when he gave people cowpox, it made them immune to the smallpox. And this is where Jenner came up with the very first vaccination and actually got its name, VACA for cow vaccine. Actually gets its name from the cow, from the Latinized root word for cow. And so anyway, this gave them a specific antibody for a specific protein on a specific virus. But as it so happens, sometimes there are viruses that have a lot of commonalities. And we are looking right now a very big hunt. And this was actually, it was a big thing when I was in graduate school. They still have not done it to my knowledge. I was researching it this morning. Haven't seen anything on it. I was watching a documentary on it as well yesterday. They were discussing the same thing. They were still trying to find a universal virus vaccine, a universal vaccine for the flu, influenza, that would hopefully give people and produce antibodies for a protein found on all flu viruses, all strains, H1N1, et cetera, the swine flus, the avian flus, all those really nasty influenzas. Now, as you look at it, we're going to talk about specific immunity for specific pathogens. Now, there are two major types. There's what's called the innate immunity. Now, what's innate immunity? You're born with it. Maybe you're born with it. Maybe it's innate immunity. Well, innate immunity, I'm going to kind of do that because it's been bugging me for years, and I always forget to do that. And I'm sorry you guys could be the ones who always experience this, but it is something that if I don't do, it's just going to bug me, and that is just how I am. And if it bugs me and I don't do it, so give me a quick to pause. Okay, I wanted to go fix it. It was bugging me to death, and I'm sorry. So, So innate immunity, you're born with it. It's because of your genetics. There are people out there who had innate immunity against particular viruses. They were born immune to it. This could be through genetics. Maybe you don't have a certain protein that the virus requires to have to dock to the cells and cause the disease. Maybe it's other things. Then there's the acquired immunity. You're not born with it. You have to be exposed to the disease or receive antibodies from somewhere else. So you have to get it. Now, this is why one of the things that always bugs me if I'm in a store and it says boost your immunity naturally. Well, there's really only two. There's innate and there's acquired. So that means either that pill or that substance they're selling you has something in there that would modify your genetics, which you probably wouldn't want to take something that would modify your genetics that could cause cancer or something else, or it's going to have the disease in it. It's going to have to be something that makes you sick, or antibodies in it potentially, but they don't. It's things that generally, unless you're deficit in, then it doesn't matter. So acquired immunity is you receive antibodies or exposed to the disease. Now, active immunity, you've been exposed to the disease, you got it, then you produce antibodies. Okay. And this is kind of where I need to boop, boop. So basically, this is when you've gotten a disease, you've had it, and you make antibodies. So if you ever get it again, your body's going to fight it. It's going to resist it. And there's artificially acquired immunity and naturally acquired immunity. Now, naturally acquired immunity is exposure, the antigen, naturally from the environment, like licking doorknobs. You put your hands on your face, near your eyes, nose, or mouth after you touch the handrail. Somebody sneezed on you. You got coughed on. You inhaled air that was carrying a virus suspended in somebody's spittle, where it aerosoled up into the air. It got kicked up from the dust. You got it that way, somehow from the environment. Or you got it through a vaccine. You can get an antigen from a vaccine. A lot of times they'll give weakened or dead, so-called dead or weakened pathogens. And they'll give you that to allow your body to fight it, to make antibodies. But in it, we also have things that will keep those on the shelves longer. We have certain preservatives. A lot of the things will add lumina-based compounds to these vaccines that are going to act as an adjuvant that will deepen the immune response, things like that. So, and then there's passive immunity. Passive, you get it from someone else very passively. They've gotten the disease, they fought it, they survived it, they give their antibodies to you without you ever having had the disease. And that is something that happens like through mother's milk or across the placenta, called naturally acquired, or antibodies as a treatment. What we have now is the ability to go in and produce an antibody in a lab in a process called recombinant antibody technology and produce antibodies. Your body makes antibodies. We used to go in and take a horse and then shoot a bacteria or a virus into them, get them to make the antibodies, and we collect the antibodies and give them. And a lot of that, like antiviral serums, we try to get the antibodies from a host who has never been, who did not die from a thing. Let's say that they were the natural carrying host, but it was not fatal for them to have COVID-19. If you can find those individuals who have those antibodies, you can actually use that to try to treat a particular strain of it. And passive immunity, like we said, artificially can be a treatment, antibody treatment, things like that. So now, something that I'm going to test you guys on this exam, you're going to see the two different kinds of histocompatibilities. The major histocompatibility complexes. And I will ask you guys these on a test. So what I'm going to do is discuss two kinds of cells. One of these cells here that I'm going to discuss, here we have a nucleus. And these will be all nucleated cells in your body, all nucleated cells, so even neurons. And then here is an antigen presenting cell. These are a lot of the times are macrophages, things like that. Okay, dendritic cells, stuff like that. Okay. So let's say we have this guy here. This guy here, what he's going to do genetically is he's going to have some DNA in here, and in his DNA he has a gene, and that gene is on a particular chromosome at a particular location, and that will allow him to produce a molecule here called a major histocompatibility complex, MHC. And it is an MHC Class 1. Now, though this cell here is nucleated, he has some DNA, and on that DNA there's a gene, and it will allow him to produce a molecule called an MHC Class 2. Now, one of the things I'm going to ask is which MHC does each cell make? One of these cells, I'm going to say which cell makes MHC1, MHC2. Okay. Now, in a minute, we're going to talk about what these MHCs do a little bit more, okay? Now, these MHCs are going to bind with certain cells. Now, Class 1 are all nucleated cells. Class 2 are found in antigen-presenting cells. Now, I want you guys to learn something here called the equals 8 rule. Equals 8 rule. Equals 8 rule is going to be this. So, we have blank times blank equals 8. Blank times blank equals 8. Now, one of these is going to be the MHC. One of these is going to be the MHC here. The other cell here, and I'm going to use purple for that, is going to be the type of T cell. Okay? So, the T cell that's here has to have a particular marker on it besides its T cell receptor. And this one here is going to have a particular marker on it besides its T cell receptor. Now, this T cell here, 1 times what equals 8? 1 times 8 equals 8. So this is called a CD8 T cell. 2 times 4 equals 8. So this is a CD4 T cell. And I'm going to ask you guys what kind of T cells interact with these, okay? CD4 versus CD8 T cells. And what kind of MHCs are made here, okay? Now, there will be a little bit more as we go along, but right now, that's what we're going to see. Now, what we then want to do is turn our attention to some other things. Now, the Class 1, that's about what happens if you're infected with a virus or bacteria that is a non-self cell, like a cell that is infected with an endogenous bacteria, virus, or like you gave me a cell, you gave me a kidney. What's going to happen here? Now, how's this going to go? And I'll draw it first before I actually go through the steps, but let's say I have a cell here. This is a cell. This could be any cell. This could be a cell here. And we could have us have a virus. And let's say this virus here is COVID-19. And COVID-19, coronavirus, gets its name because of the shape. Looks like a little crown. And inside this virus, there's going to be some DNA. Okay, there's going to be some DNA. Hopefully not RNA. That's a really, really bad one. Okay. Now, this cell here, remember, has a nucleus. Okay, nucleus is found here. Now what's going to happen is this virus is going to dock. It's going to get docked here. It's going to dock in. When it docks, it's going to slip some DNA into a cell, and this DNA is going to end up going into your nucleus, and it's going to hijack it. It hijacks your cell's transcription and translation machinery. Okay, so that's that viral DNA. Hopefully not RNA, because those are really bad things. RNA-based viruses like Lassa, Ebola, things like that tend to be really deadly things, really nasty things. So this virus DNA, viral DNA, is it locks and it shoots into our body gets it in there. It's then going to do something interesting. There's one of two things that's going to happen. It hijacks our cell. It can make its DNA part of our cell, and that's what happens with chicken pox, why you get shingles, and you have shingles, you can get it for the rest of your life, or why you have herpes forever. But if you get a typical rhinovirus, cold virus, or some COVID-19, it just kills the cells, they burst, they die, and they move on. So it's going to come in, it's going to start to make your cells start to produce viral pieces of virus, and it's going to start to assemble virus pieces, and we're going to just refer to these as endogenous proteins. Okay, endogenous proteins. Now, these endogenous proteins that are found here, what's going to happen is, let me make sure this thing stays focused, is your cell, remember we got DNA in here, and there's a gene on that DNA somewhere, and that gene, let's say this gene says, let's make my MHC, and we grab one of these endogenous proteins. Now, because this is a nucleated cell, this is an MHC1 molecule, and it's going to put on its surface, it's going to grab one of these endogenous proteins, and we will now say that it is displayed. It's displayed. It's putting it on display. It's going to be like waving a flag in front of a bull. Now, right here, we have a T cell. Now remember, this T cell has T cell receptor, TCR. It has a CD8 T cell, CD8. 1 times 8 equals 8, CD8 T cell. And this CD8 T cell is going to be able to bind, and it's going to recognize this antigen. It's going to recognize this antigen, this endogenous protein, this antigen, okay? And this antigen that it recognizes, what's that going to do? Well, now that it does this, it's like a red flag for a bull. And it puts it out there, and this CD8 T cell will now be able to attack and destroy. And what's going to happen is the CD8 T cell, he's going to become cytotoxic T cell. And he is going to attack and kill the cell. Okay? He will attack it. And again, the needs of the many outweigh the needs of the few. This cell here, if we let this virus replicate, it will go out and spread out. So one of the things, too, remember, not only is this cell being infected, but he's already making alpha interferon. And he's trying to warn the other cells to arms. Okay, to arms, to arms. The virus is coming. The virus is coming. To arms, to arms. The virus is coming. Okay. So he's already releasing out alpha interferon, but what's happening is, guys, he is our cytotoxic T cell will come in and destroy this cell, hopefully preventing it from making more viruses, because once it assembles them, and if it ruptures, it could fill hundreds of thousands of viruses to each all go in and infect other cells. And as this starts to happen, this is where the disease comes from. This is why the sore throat sometimes. This is why the respiratory respiratory issues. This is why the kidney issues. This is why the hemorrhagic fever, the bleeding out, crashing and bleeding out of hemorrhagic diseases, Marburg and Lassa and nasty things like that, why they're able to do these things, okay, is so our goal here, guys. Now, here's what's scary about COVID-19 is that COVID-19 can actually replicate inside of cells much warmer than normal coronaviruses. COVID-19 can go into the lungs and replicate where it's generally too warm for most coronavirus. Most coronavirus is going to be up in the head. This can go down further into the lungs. And that's one of the scary things about it. Fortunately, the mortality rate is very low for COVID-19. Most people who have actually died have been healthcare workers, though, because they're in there working with the patients. And if you treat them with fluids and antivirals and a variety of things, you might be able to help them, interferons, stuff like that. All right. So now that it has been taken care of, we got rid of it. Okay. Now, our cytotoxic T cells. So as we see here, we have a virus. This virus, even a bacterial pathogen, there will be some abnormal endogenous proteins. Abnormal proteins will show up. Whether it is viral proteins that the cell has been making, or bacterial proteins, whatever. There are bacteria that like anthrax that like to go inside. That's why we give ciprofloxacin for certain things. Ciprofloxacin works really well in endogenous bacterial infection. You go in, these endogenous proteins, abnormal proteins in the cytoplasm show up. Or MHC1 grabs it up, and like a Hail Mary, it grabs one of them. Now, there could be many different ones. It grabs it and displays it onto the outer cell membrane of this infected cell. And then what happens is our CD8 T cells come in. They can become various things like cytotoxic T cell. That's what TC, kind of cytotoxic. But it also may become a memory T cell. And memory T cell will sit around and remember. And it remembers that virus it saw. So let's say we get around this virus again. Identical virus that we've gotten. Now, everybody's like, well, how do I get cold every year? Well, there is so many strains of the cold. Innumerable strains of the cold. There's innumerable strains of the flu, influenza. It is just so hard. And there's so many. You never get the same cold twice unless you have some serious problems. So a lot of times, well, I got sick again. Well, a lot of that secondary infections, bacterial infections, secondary to viral, or even secondary to allergy. So, all right. So what we're going to see is, guys, is now the other half of this is what happens with antigen-presenting cells. Now, antigen-presenting cells, I'll talk about this. I'm going to start off by drawing it, but I urge you to draw this with me. I want to make sure you understand what's going on, not just memorize your way through things. So we're going to start off here. Our little story is going to be like a Cinderella story. It's going to be a Cinderella story. Now, in our Cinderella story, we have a Prince Charming. That stands for antigen-presenting cell. And then we need to have our bacteria, don't we? We need to have Cinderella. Doesn't Cinderella sound like a disease that you might get? Now, Cinderella, this bacterium here, has various antigens on its surface. And there are antigens inside. There's internal proteins. There's external proteins. Let's use these external proteins. Let's draw an X, a circle, a square, and let's draw a triangle-shaped antigen. Let's draw these antigens. Now, this is a bacteria. Let's say this is Cinderella. Now, Cinderella is made up for the ball. Her fairy godmother had given her all these lovely antigens. And what's going to happen is she is going to find her Prince Charming at the ball. And Prince Charming brings her in. Remember, this is called phagocytosis. And he has her in a phagosome. where they're dancing. So that's them dancing, okay? So here we have her and her Prince Charming. They are dancing together at the ball, and Cinderella's got all of her beautiful stuff her fairy godmother gave her to wear, her gown and her slippers and the necklace and all the beautiful jewelry that she gave her. Now remember, there is also a nucleus here as well. Now what's going to happen is this cell, this antigen-presenting cell, did phagocytosis. Now, since this is a Disney story, we got to put two of them on there, make it look like Mickey Mouse, right? What is this? You're like, what is he going on with? This is a lysosome. And remember, lysosomes, they're full of carbohydratases, proteases, lipases, nucleases, and acids, enzymes and acids. So they have ACEs. And these ACEs go out, these enzymes go out, and they're going to start to eat up this bacteria, eat it up. And what's going to be happening, now I can't draw it here, is all of these antigens are going to be left behind because we ate up the bacteria, we ate it up, and in our DNA here, we're going to produce an MHC. And let's just grab this one. Let's grab square. And it grabs square antigen. Now, do you agree there are other antigens? There was also circle, X, and triangle, okay? And this was an MHC2 molecule, and it's displaying the antigen. The antigen is said to be displayed. Now, we know that it's got to talk to a CD4 T cell. Now, there's several CD4 T cells out in the land. These are all CD4, CD4, CD4. These are all CD4 T cells. Now, some CD4 T cells, now remember, they all have T cell receptor. I want to talk about they all have that. They also have that CD8 marker that makes them. But what's interesting is we have some different ones, different T cells, okay, with different T cell receptors on them. Okay, now if this is where Cinderella's story comes in, okay, so think of the handsome prince, Cinderella, she dropped her slipper. Think of the displayed antigen is her glass slipper, okay, that's the glass slipper. And what did he have to do? He had to take this around to all the ladies in the land to see if it fit. And he's like, no, that doesn't fit. That T cell receptor don't fit. Oh, that doesn't fit. Oh, that doesn't fit. But finally, he finds one that does fit. And that's it. That's Cinderella. That's his Cinderella. And he's found his Cinderella. He's found his CD4 T cell who matches his antigen. The shoe fits. That's it. That's the girl that he's meant to be with. That's the girl of his dreams. Imagine that being your criteria. How many women have the same shoe size? How many women right now listening to my lecture at this moment has a best friend with the exact same shoe size as you, and you guys share shoes with each other? Like, oh, girlfriend, just borrow those, bring them back. I know you're going out to the club. You want to look good for the men. Borrow my shoes, okay? But what's happened is Prince Charming here, has found his Cinderella. So what does he got to do? He's got to display. Now, here's the thing about CD4 T cells. They are very picky. See, CD4 T cells, with their antigen, though they go with it, they're a little bit like Beyonce. They're picky. They're like, if you like it, then you should have put an MHC on it. If you like it, then you should have put an MHC on it. The MHC2 has to be on it. If you like it, you should have put an MHC on it. You can't, this CD4 T cell, if you tried to propose to this woman here without displaying it, if you didn't get on your knees and get down and hold up that and open that box up, if you didn't go to Jared, if you didn't give her her ring the way she wants, she's going to not see it. And so this guy here, if you like it, then you should have put an MHC on it. T cells can only see antigens when they're on MHC2s. Okay, so if you like it, then you should have put an MHC on it. If you like it, then you should have put an MHC on it. Okay, so now that he's found out, what's going to happen to these two? Well, they're going to sweet talk. They're going to sweet talk. Oh, I love you. Oh, I love you. Marry me. Be with me forever. Live in my castle. I love you. No, I love you. I love you. No, I love you. And he's like, be mine. Marry me forever and always. Happily ever after the end. All that good stuff. And we call this co-stimulation. They use some molecules here. Now, if we can prevent these interactions from happening, we have we have immune suppressor drugs. Okay, this is why I'm teaching you this, so you understand immunosuppressants. Okay, now, with that in play, we now have activated our CD4 T cells. Now, so what's happening here is that you engulf it. It has multiple antigens. They break them down with their lysosomes, and what happens is the antigens are left behind, and the antigen proteins are grabbed up by an MHC2, displaying it. Then that displays presenting to antigen-presenting cells, to CD4 T cells. And the so as you can see this, the CD4 T cells, they're inactivated. They have MHC, they have T cell receptors, and there's very specific binding sites for the antigen, but it only occurs when there's a matching one. Now, when I drew 4 different ones, there was a T cell for every antigen on Cinderella, but only one can be activated. So we got to co-stimulate. That's the sweet talk. Now, ladies in the room, listening to me now, ladies, when your man finally pops the question, he gets down on that one knee, and he asks you to marry him, I know what's going to happen. After you say, well, if you say yes, you go, yes, yes, 1,000 times yes. Who's the first person you're going to call? Now, guys, those ladies, they all said mama. They're going to call mama. They're going to call mama. And so you're either going to have, she's going to be excited, and she's going to never forget it. You know that ladies, they're going to get very excited. They're going to get very excited. They're going to call mama, and they're never going to forget when you asked them to marry, when you proposed. So make it memorable, because they're never going to forget it. So make it good. Now, what's going to happen is this might become cytotoxic or memory T cells, okay? And so we're going to go out here and take this memory T cell. And also what might happen, cytotoxic or memory. Now, that's if it answers quickly. If it's very slow, it's going to be helpers and suppressors. And that's what I want to draw here. Okay, now that is fast. What if it responds slow? Then you become helpers or suppressors. Okay, so ladies, they're going to call mom. Call mom. And that's one of the things that happens. I was trying to get one here. She might say, I don't know, husband shame. So I've got call mom. That's one thing she's going to do is call mom, cytotoxic or memory T cell. Husband, or she's going to have husband shame. She doesn't want to tell anybody who she's marrying, husband shame. Okay, so I just come up with that one on the spot, guys. That's not too bad. I've always loved my mnemonics. So what we've got now is we're going to take one of those helper cells in a minute and follow them along. Okay, so we're going to use the backside of this, and we're going to do that in a moment. Now, so there is a difference. If it is activated very quickly, responds very quickly, it becomes cytotoxic or a memory cell. If it responds slowly, it becomes a helper or suppressor. So think about this. If she knows she's going to marry you, she's going to be like, yes, yes, no questions about it. Let me call mom. If she's not sure about it, she might have husband shame. She's like, do I really want to be married with this guy? She might have husband shame, helper suppressor. Okay. Now, so here we have the foreign antigen had shown up in an antigen presenting cell. MHC2 had come in, class 2 MHC, displayed it to a CD4's T cell receptor. There's the CD4 protein, why it's called a CD4 protein. Here is a CD8 protein, so I call it a CD8 cell. All right, and what's going to happen is you now turn that cell on. Now, if it's a cytotoxic T cell, it can release perforins and it can destroy the cell membrane. Remember, perforins is when you stab a cell. It can release cytokines, and that can trigger apoptosis or secretes lymphotoxin. If you guys do not know what apoptosis is, this is something I teach in A&P 1. This is programmed cell death called apoptosis. Okay. Now remember here, help me catch some. Helpers, help me catch some. Now, activation of a CD4, usually it's going to be a helper T, memory, or suppressor, or cytotoxic, which I could also put help me catch some, which I need to add to this as well, and that's kind of my fault for leaving that. And I will discuss, all you got to do is put cytotoxic here, and I need to do that in my notes, and I'm going to kind of do that now just to remind myself I need to do it, because there's always things that I feel like I need to put in notes that I leave out sometimes, not purposeful, but accidental. So if it's an activated T cell, it secretes cytokines. Memory T cells, they just remember, they remain in reserve. Whenever you make cytotoxics, you also make memories. Remember, that is your, when you call mom immediately after he asks you, you say yes. You call mom, he said yes. I said yes. It's awesome. And they stay in circulation, and anytime you ever see that antigen again, they become cytotoxics immediately. So the cytotoxics aren't produced by directly activated CD4 T cells immediately after activation. They are produced from memory T cells. So I kind of have it here, but I kind of don't have it here, and I was trying to be more accurate. Let me know if you think it needs to be this way or the way that I have it originally. If it makes more sense to the student. From my end of things, I thought it made more sense 1, 2, 3, because I did have cytotoxic. I just want you to know they are made. Okay. Now, the suppressor T cells, they produce suppression factors. They make sure that other T cells and B cells are not turned on. After the immune response has initially responded, this is like after a bad accident has happened. The suppressor T cells, like when the cops come out and says, "Move along, everyone, nothing to see here," and they go on, right? They limit the immune reaction to a very single stimulus. We don't want to keep making the immune system fire when it doesn't need to. It's like, okay, it's like the Winter Soldier's been thawed out. He did the thing. Now we got to put him back and freeze him again, deactivate him. Wait till we activate him and give him his codes. Let him do his mission, put him back in freeze, deactivate and put him back in freeze. And that's kind of what's happening here, okay? You just, we want to make sure that we put it up and we don't keep the super soldier suppressor T cells out. We want to make sure they inhibit things. They prevent the spread of your T cells and B cells from activating unnecessarily. When that happens, we call that autoimmune disease, and we don't want that. Now, B cells here, they are going to be kind of like mama. And this is like mama. This is the mother So let's say, guys, I take this guy here, okay? So let's say I've got a helper T cell here. This is one of my helper Ts from before, activated to square antigen. Okay, it's activated to this. It's activated to square antigen. Okay, not X, not triangle, not circle, square. Okay, it's activated to the square antigen. Okay, now it's a helper. Now remember also we have B cells. Now this B cell, what B cell has got? It has what is called an IgD. The IgD is the notorious BIG. You down with IgD? Yeah, you know me. That's not OPP. Don't ask me what OPP means if you guys don't know. That's from my day, and that's a very dirty thing. Okay, so you down with IgD? Yeah, you know me because it's from the notorious BIG. Dang it, B cell, get busy. The IgD here is going to serve as an antigen receptor. And this cell here has been stimulated to square antigen. And let's say square antigen shows up in my blood. And it comes in and it binds to this. See, we have many B cells who have a particular IgD who have binding sites for a particular antigen. And again, we make IgDs for antigens you never seen before. Literally, you've got a genetic slot machine in the cells that make these antibodies, and it's like, oh, let's make a new batch of antibodies. Ka-ching, and spin the wheel. Okay, there we go. Here's what we got on this slot machine. Make some of these. And then it'll do it again, and it makes a bunch of different IgDs. So this one here has a specific IgD that can bind to square antigen. So we will take this square antigen in, we will internalize this antigen, and this will literally rewrite the DNA of this. And then what we're going to do is we are going to then produce an MHC2 molecule with that antigen on it. And we now say it has been sensitized. Not activated, sensitized. Now you're probably wondering, now here's the thing. What's this like? If B cells like mama, this is the family. Now before Prince Charming asked her to marry him, he'd already went over and said, hey, Mom, I love your daughter. I respect her. She's a special lady to me. She's the most special, most important woman in the world to me. I love her. I would like to ask your permission if I could marry her. And she says, yes, I'll give you my permission. Yes. Here is a ring that I have. You could propose to her with that. So she's now been sensitized. She knows it's happening. Mama B cell knows that her daughter is going to be asked to marry her APC, her Prince Charming. Antigen presenting cell is going to co-stimulate her. She knows it's going to happen. So think about it like that. So it's been sensitized. Now, daughter had come in and says, Mama, he asked, I said yes. She has to act like she doesn't know that's going to happen. So what's going to happen? She will become a plasma cell, and that will make antibodies, or she will become a memory B cell, and she won't forget. Because mama will never forget the day her daughter called her to say, I'm getting married. Now, the antibodies being made from a plasma cell. Now, what are these antibodies? These antibodies, guys, they're going to be anti-square antibodies, not anti-antigen triangle, not anti-X, not anti-circle. Now, let me tell you something really cool. Let's say, if we will, here's Cinderella. Her cousin, let's do it over here. Her cousin, Rendercella, shows up. Now, Rendercella has that one, but she also has half moon, star, curlicue on her. Those antigens, weird antigens, but she's got square antigen too. This mama B cell could produce antibodies, antibodies against that. And they will attack even this one. It will attack Rendercella bacteria because it's got the same proteins as Cinderella bacteria. So if this was two closely related species of bacteria, say like two different strains of E. coli, then your body could fight it off. And this is how cowpox, which is not very harmful to us, was able to give us antibodies against smallpox, which is very deadly. Okay? And I hope that makes sense because B cells can recognize the antigen while it's on the bacteria or floating around in its native form. T cells can only recognize them when they are on an MHC, because if you like it, then you should have put an MHC on it. Okay, I hope these drawings make sense, because this is a big concept that if you can understand, lots of things are going to make more sense. Okay, so we take in that antigen, we process it, and then we put it on MHC2 on the B cell surface. That is the sensitization process. Once sensitized, so how does it get sensitized? Well, there was an IgD, that is the receptor for the antigen. It's on there's millions of B cells, and they may have one particular antibody. So you might have, let's say, 100 that have one antibody, another 100 have another antibody, another 100 have another antibody. I'm just spitballing numbers. It really, it's like slot machines in Vegas. It's like you make some, you randomize that section of the gene. It's pretty cool. Now, this gives us flexibility. Now, if that antigen ever shows up, whether it is on the surface of bacteria or floating around in the blood, a B cell will possesses MHC2, and when you sensitize it and the antigens engulf, it binds it, and it's sensitized to it, and then a helper T cell activates it to make antibodies. Okay? Now, you can attack these antibodies there. Now, they'll make memory B cells or plasma cells. They produce an antibody for a particular antigen. Memory B cells, they can be in there for like 4 years or so, just waiting in reserve if I ever get that infection again. B cells, remember, native form. That means it could be floating around. It could be on the surface of bacteria. We can activate it anyway. T cells can recognize the antigen if they are displayed only on an MHC. So here you can see I have some antigens that show up floating around, or it could be on the surface of bacteria. We sensitize the B cell. Now, only can this right here. Here's the deal. Yeah, this can happen. This can sensitize it, either floating or here. But I have to activate this CD4 to a helper. The helper must then come in and activate activation of the B cell can lead to the plasma cell memory B cell production. Okay? Does this make sense? I hope. And eventually that's what happens. Okay. Now, B cells and T cells. T cells, what do they talk to B cells? We already know that. They say, help me catch some. B cells says, my pleasure. because T cells come in helper, memory, cytotoxic, and suppressor. B cells come in memory B cells and plasma cells. T cells, B cells. All right, so these are our acquired immunities. Now, with acquired immunity, we made antibodies. And all antibodies, what does antibodies have? Antibodies have some different types of structure to them. And I want to talk about antibody structure shortly here, briefly. And I've got to find a piece of paper I can draw on real fast. Now, antibodies, they have two major components. They have what is called a heavy chain, and they have light chains. Okay, now, as you look at these, let's use blue for heavy. And then I'm just going to use this color. Those are just some disulfide bridges. You're like, what's a disulfide bridge? You got to take me for general biology to learn that. And then we're going to have our light chain. And that's on an antibody. Now, antibodies have what is called a constant region that never changes. It's got some sites on it. For example, complement binding sites and macrophage binding site. And over here, we have what is called a variable region. And on the variable region, we have this space right here that is the antigen binding site. That binds to the antigen, okay? So what you're going to see here is, guys, your basic antibody structure allows this to be highly variable because it could be different for every time we make one, and the constant region for a particular type is always the same. So we have same and we have different. Okay. Now, in the variable region, there is a set of light chains and a set of heavy chains, but in the constant region, there is only heavy chains. Okay. There's only heavy chains. Now, there's a macrophage binding site and a complement binding site, which are part of non-specific defenses. Now, antibodies, think about it, there's an old lady named MADGE. MADGE. IgM, IgA, IgD, IgG, IgE. Now, IgM, that's the main attacker. These are the ones found in blood. These are your blood type antibodies. They are the first to attack, found in the blood, and they exist as a pentamer. What do we mean? 5-body collection. They're a pentamer. Mer. IgM is a pentamer. Mer. Main attacker. IgAs, they attack before they enter the body. They're in like tears and saliva. They are dimers. They are a dimer. A dimer. IgA is a dimer. They attack before they enter. IgDs, they're on the B cell surface. Remember you down with IgD? Yeah, you know me. That's from the notorious BIG. And they play a role in B cell sensitization. That's a hard word to say, sensitization. And I like to say, dang it, B cell, get busy. The D in dang it, B cell, get busy lets you know that IgD is on the B cell surface. The A in attack before they enter lets you know what the IgA does. The main attacker, IgM, first to attack, tells you what it does. IgGs, go get them. They're the ones that fight the microbes. These are the most diverse because every time you get a virus or bacteria, fungal infection, protozoan infection, whatever infection, you get coronavirus, your body is going to make a particular IgG against that strain of the flu, against that strain of a coronavirus, against that strain of rhinovirus, against Ebola. Hopefully you don't, because Ebola's got a 60-some% mortality rate. IgE binds to your mast cells. It makes your allergic responses happen. E2, E2. And these help you remember what each one does. Okay? So here's your different classes of antibodies. IgM is a pentamer, IgA is a dimer, IgG, IgE, IgD. Now, how do you classify these? We use their their constant region, to actually describe the antibodies class. It's based on their constant segment of the heavy chains. Now, the antigen binds to the antibody at what is called to its antigenic determinant. The antigen the body recognizes, so an antigenic determinant is an antigen that an antibody's antigen binding site can bind to. When they do, there is antigen-antibody complex. Now, this is why I say we hope it's an external antigen and not one inside, because this bacteria right here, we're not going to get those antibodies inside of it. And if we can't get it inside, we're not going to get it to attack very well at all. If it's on the inside, it's not going to happen unless it's an external antigen. So every time we do that drawing that I did, these drawings here, there's a chance that when I display this, that antigen is inside this bacteria and useless. And it's a chance it's an outside one. It's a chance that maybe this one here is also found in other members of that species. And if my body's seen it, for example, here, we were talking about Cinderella. Well, if Rendercella shows up, and Rendercella, not Cinderella, also has square antigen, like we were showing, then my cytotoxic T cell is going to go, bam, get rid of you, kill you, bam. If it shows up here, we can sensitize a B cell, and an activated helper T cell can help produce antibodies. So that's important that we understand. And then it's important that we understand these antibodies and what's going on, and how that if we make an antibody sensitized to an external antigen, to an antigenic determinant site, then we've got something good. And we have what's called the antigen-antibody complex, and hydrogen bonds lock the antigen to its antigen. Antibody to its antigens. Now, what do antibodies do? Antibodies, one of the things they do is they neutralize viruses and bacteria. They make it incapable to bind. If they can't bind, they can't get on the surface and cause the diseases. See, it is basically what happens is this is another thing kind of about coronavirus, is you can have it, you can sneeze and give it to people when you're not even asymptomatic. Because you don't have the symptoms of the disease until the virus goes in. We say you have the disease when the virus goes in, starts to destroy cells, and that causes damage to the tissues, like the lungs and things like that. And then that's when we say you have the disease. But you can also spread coronavirus without being diseased yet, or bacteria. Now, if we can make it so they cannot bind to cause the damage, whether they're going into the cell or they're damaging, releasing cell toxins from the bacteria, then they can't do it. They do precipitation and agglutination, where these antigenic determinants, there's usually more than one, and multiple antibodies stick all over it, and they make what's called a precipitation when there's an immune complex. When antibodies are bound to multiple antigenic determinants, called an immune complex or precipitation. Now, if it forms big ones where multiple cells stick together, it's a clump called an agglutination. Then these antibodies that are bound, antibodies have the ability to bind a complement. And activating complement binding sites of the antibodies, they're activated when the antibody binds to its antigen. So that can actually activate furthermore complement proteins. They can attract phagocytes, like sharks with blood in the water. Neutrophils, eosinophils, monocytes, nymphagocytes. Opsonization. That is, the name opsonization means that if we coat it with antibodies and complement, we allow phagocytosis to happen easier. Think about a bacteria is like trying to grab a bar of soap with wet hands. They're slimy and covered in gooey, sugary glycocalyx. And if you try to grab them, phagocytose them, it's just going to squirt away. But if you cover it in antibodies and complement proteins, it's kind of like you put sand on it. And now we can grip it. And that's going to enhance phagocytosis. We can simulate inflammation by taking basophils and mast cells that are in the area. When antibodies bind to mast cells, and they also bind to basophils to increase inflammation. They also prevent bacterial and viral adhesion to tissues. Now, that's a little bit like neutralization in a way, is a little bit like that. But it's coating the antibodies on it. Sometimes there's other reasons why antibodies may neutralize, make it incapable. It can lyse it, it can damage it, it can destroy the docking proteins, but just coating it makes it incapable of attaching as well. So neutralization and prevention of viral bacterial adhesion are a little bit similar, but not perfectly identical. Okay, and that's why I'm going to parse these out. Now, I do want to talk about primary and secondary immune response. When you first get exposed, you get a primary response. So, like if you ever get an infection, a virus, first time, your body produces what we call the primary response. It takes a while to get it going. You got to take time to make antibodies, so it takes you a while to get over it. If you get it again, you fight it quicker. The secondary immune response is more extensive and prolonged because the primary had already primed memory cells. You already have memory cells. And this is what vaccinations do, is they help keep the circulating antibody titer and memory cell counts up. This is why you need boosters. Now, what is the primary response? It takes time. This gets your antibody titer, or how many antibodies you have up. This takes time to develop because antigens have to eventually get the T cells to the B cells. B cells have to make plasma cells. Plasma cells have to make antibodies. Antibodies have to arrive. And it can take 2 weeks to develop, and then it's gone away very fast. And it's actually, we make IgMs faster than we do IgGs, but the IgMs are less effective. But once I've done this, I have memory B cells. Secondary immune response involves activating already activated sensitized B cells, who are memory B cells. This means, and it even requires less antigens. The antibodies are quickly produced, and it's the good IgGs. And those IgGs can be made longer and quicker. They're more effective than IgMs. Okay? And this is what basically a vaccination does for you, is it ensures that you're producing a secondary response even if you see something. There's been some evidence to suggest even having a flu vaccine may in fact help you against coronavirus. Not sure yet. I remember I was in graduate school, I sat in on some, we had an awesome seminar from the Center for Disease Control, piggybacking with Quillen College of Medicine, and I got to talk to some of the people there at CDC making vaccines, and they were talking about how they were trying to do research. Now, private companies are trying to get that done too, and they might be making more headway, kind of like SpaceX or Tesla like company, but now this will be biotherapeutic companies, private like that. So pretty cool stuff. So you could see the IgMs are first and the IgGs are the more effective, and basically secondary, you make IgGs all day long and you make them a lot. It takes about 2 weeks to get your IgG levels up with the primary response. Secondary, you've already gotten it the first time. Now it's your second exposure and you're really quick to fight it. Now, allergies, they are immune responses. They are excessive, inappropriate responses to something that's an antigen. Like here we have pollen. Pollen is a very common allergen. And there are proteins on pollen, like sporopollenin, things like that. And these proteins set off immune responses. So if it's an antigen that sets off an immune response that are called allergies, we call them an allergen. And you can get it from being exposed to them too much or too little. But once you get those antigens, you take IgE production from a plasma cell, and the plasma cell's IgEs bind to your mast cells or your basophils. Remember, bad on you feels, basophils, or on your mast cells, and they'll degranulate, releasing things like prostaglandins for pain and inflammation, things like that. This is why you get runny noses, allergic rhinitis, the pollen allergies. You start doing the pollen salute. And if we have that mixed with coronavirus outbreaks, it might become more likely people are rubbing their eyes and rubbing their face to get coronavirus. So that might be a good time to have that. It's here in the county that I'm in right now. So that's scary. So what we're going to see, guys, we are done. And this is by far this and the chapter on digestion are the two most difficult. I think this is the most difficult of the three. It's the most important of the three. Digestion is kind of dry. Hardest thing on that's the hormones that regulate digestion, but this is a big topic. I want to make sure I help you understand it. I wouldn't use the analogies I do and try to teach you as enthusiastically as I do if I didn't think you could do it and didn't know that it was important. I did share on my lecture page my AMP1 notes. If you guys do need those, I recommend you use them because I think my notes a lot of times are more in detail than some of the other professors' notes. So I did give you guys my AMP1 notes as well. And this is the stuff for here for lymphatics. I'll have the study guides at the back for lab. I'll get into that more. But this is exam 3 material, so this will be after you guys finish this exam, and you should have your grades by the time you see this lecture anyway. So, guys, thank you so much, and I am done for the day. So we'll be on to the next chapter. Thank you guys so much.